Welcome back to this last lecture of the introduction module. I'm Cédric Notre-Dame, a researcher in bioinformatics at the Center for Genomic Regulation in Barcelona. Today, the topic will be a little bit special because I will tell you about multiple sequence alignments, a subject that has been my main theme of research over the last 20 years. But let us pick up where we left things. Last time, we discussed about how to gather sequences homologous to your sequence of interest using BLAST. At that point, I was focusing on pairwise alignments of two sequences, and how useful they were. Well, today I will show you that in many circumstances, it can be very useful to compare more than two sequences. This is especially true for phylogenetic and domain analysis, two of the many applications of multiple sequence modeling. We will finish by discussing how these alignments can be very useful to analyze exome data and how this analysis can lead to therapeutic choices while taking into account metabolic pathway information. Let us start with multiple sequence alignments. Given a set of sequences having diverged from a common ancestor, a multiple sequence alignment is a table in which every column contains residues that were homologous in their last unique common ancestor. For instance, if you look at the alignment shown here, the column with a conserved W, tryptophan, is a statement that if we are to believe this model, the common ancestor of these sequences had a tryptophan on this position. If a specific sequence has lost a residue, the corresponding location will be padded by a dash symbol, which is referenced as a gap. A gap reflects either a deletion from the ancestral sequence or an insertion in other family members. Why is this representation of the sequences so useful? Well, let me start with a small story that is actually true. During the Second World War, a British engineer, Abraham Wald, was asked to help improve the survivability of the warplanes. To that effect, he did something very smart. He started monitoring the bullet impact on the squadron planes when they were coming back from mission. For instance, the figure I'm showing now will be the summary of all the impacts received by all the aircrafts of a squadron during a period of time, a month maybe. When shown this picture, most people, and I did, argue that the information it contains should be used to protect the parts that are the most exposed to the impact. Of course, that will be too simple, and the stroke of genius of Weld was to figure out that one needs to do the exact opposite. Indeed, if you see a bullet impact, it is because the plane was hit and came back. You therefore need to protect the patches that are suspiciously depleted from the impact, most likely because any plane hit on these spots did not make it back. The analogy with multiple sequence alignment is very strong. In an alignment, every sequence is like a plane in a squadron, and each mutation is like a bullet impact. The reason why some positions, like this column of W, never seem to be mutated is not because they are spared from mutation. On the contrary, it is because any time this position was mutated, the organism carrying this mutation did not survive. In many ways, our multiple sequence alignment can be seen as a collection of real-life experiments carried out by nature, and whose outcome is revealed to us through the multiple sequence alignment process. The first and most obvious application of multiple sequence alignment is phylogeny reconstruction. Phylogenetic trees can be very useful when dealing with infectious disease. For instance, whenever a new bacterial germ is identified and sequenced, it's usually difficult or even impossible to cultivate it. But one can use phylogenetic reconstruction to place this organism on the big tree of life. Doing so can yield very important clues with respect to the biology of this organism. This is exactly what Didier Raoult did at the Timon Hospital in the early days of genomics. His team sequenced Tropherima whippley, an intracellular parasite that was very difficult to cultivate and therefore very hard to diagnose. And they therefore did a very large-scale phylogenetic analysis to figure out which organism was the closest and what were the missing metabolic pathways preventing these parasites from being grown in a simple medium. Thanks to this analysis, they could come out with a suitable medium that led to a cost-effective test allowing this deadly bacterium to be effectively treated. Phylogeny is not only useful to tell the relationship between species. It can also be used to figure out the relationship between genes. For instance, our human genome contains about 500 protein kinases. These proteins are very important because they regulate almost every aspect of our biology, from the cell cycle all the way to the triggering of immune reactions. In order to figure out which kinase is involved in which biological process, it is very important to be able to classify them. This is just what the company Seugene did a few years ago with the kinome complement of the human genome. This classification is very important and useful because it tells us about kinases similarity. If you have a drug meant to target a specific kinase, this classification will let you know what will be the most likely off-target effect, meaning a target that was not initially intended. Computing multiple sequence alignment and phylogenetic trees is not a simple task. And even a rough overview of the methods involved will be well beyond the scope of this introduction. What you should know is that the computation of an alignment is what computer scientists call an NP-complete problem. It means that it cannot be done exactly. It can only be done approximately. This is the reason why so many methods are available for multiple sequence alignments. The most widely used are Clustal, MAFT, Muscle, T-Coffee, but there are many others. In fact, it turned out that the 10th most cited paper of all time is the original publication of the Clustal multiple sequence aligner. This should give you an idea of how important these methods have become in biology. Good alignments are essential prerequisite for phylogenetic tree computation. But the problem of tree computation is even more complex than multiple alignment computation. PhyML and RaxML are among the most widely used software for this task. All of these methods are freely available, and all you need to run them is a good computer, preferably running Linux or a Mac which runs Linux as well, because most of these programs have been developed to run on Linux. For the less technology inclined, it is also possible to run these programs over the internet, just like we've seen for BLAST. In this case, you simply need to cut and paste your sequences and launch the computation on the host machine from any computer you have access to. It can be a Mac, Windows, or Linux. Here, you can see, for instance, the T-Coffee server where such computation takes place. In a similar fashion, most phylogenetic methods are available online, as shown here with the very popular site ngsphylogeny.fr. Because they indicate the expected level of conservation of every position on a protein family, multiple sequence alignments are very useful to identify putative function in protein. This is the reason why the relative level of conservation of residues has been used to identify sequence signatures that make it possible to recognize protein domains in proteins otherwise uncharacterized. Domains are extremely useful to understand the biological function of a protein. For instance, let us compare BRCA1 with PFAM. PFAM is a database that contains a signature of over 16,000 protein domains. Each of these signatures was established from a multiple sequence alignment. As expected, we find the BRCA domain but we also see a zinc finger motif that suggests a mode of action. And we also find the BRCT domain that are shared with a large number of non-BRCA1 proteins that are otherwise involved in DNA damage repair. Domains discovery is not the only application of multiple sequence alignments. More recently, these alignments have started being used in order to identify potentially deleterious somatic mutations, that is to say, mutations acquired during our lifetime. Indeed, when doing a multiple species MSA, one gets a lot of information about highly conserved residues. This knowledge can be very useful for cancer diagnostic. In practice, this is done using a sequencing technique known as exome sequencing. Thanks to the dropping cost of sequencing, it is now becoming routine in many hospitals to sequence all the coding regions of a tumor. This technique produces a sequence of every protein in the genome. And when these proteins are compared with the original genome, the non-mutated one can identify all the mutations contained in the tumor. But how can you tell which mutations are harmful? All these mutations are like too many needles in a haystack when all you need is one needle. Given what we have just said about evolution, it makes sense to assume that any mutation affecting a highly conserved position can be considered a good candidate for being a tumor promoter. This is exactly the principle behind methods like SIFT that use multiple sequence alignment to predict the potential toxicity of every mutation found in an exome sequencing experiment. Unfortunately, all cancer-driving mutations do not affect actionable proteins. In other words, a drug is not available to target each and every protein that exome sequencing may reveal as relevant for a given patient. That's bad news, but there is good news. And systems biology can provide powerful alternative approaches. For instance, this graph shows all the known pathways associated with prostate cancer and some of the drugs able to target each of these pathways. If your exome sequencing suggests the involvement of a protein in any of these pathways, then you're in luck and you may target the whole pathways regardless of whether the drug hits your protein or another protein that will be upstream or downstream of your target. Now, of course, you will need to know in which pathway of interest your protein is involved. In order to ask this question, the most popular resource is KEGG, the Kyoto Encyclopedia of Genes and Genomes. Pathways are very complex entities. It is important to realize that they bring our understanding of protein function one step above. With a pathway, you do not merely know what is the biochemical function of your protein, you also know in which context this biochemical function is being carried out. It's a bit like if I were describing you as a medic by training or describing you as someone working in an oncology department. These two layers of information somehow contribute in a complementary way to the definition of your function. Of course, the definition of a protein function is just as complex as the definition of the social function of an individual. We are often tempted to define proteins in relation to the disease they cause when they mutate. But a single protein can be mutated in very many different ways and therefore involved in many diseases. Even when it is not mutated, the same protein may be involved in different processes, just like you may work in different hospitals and even occasionally stop being a doctor, for instance, when coming home and doing homework with your kids or simple housework. In the protein world, the right way to dissect all of these things is called a gene ontology. It is a structure of vocabulary that provides a non-ambiguous way of naming molecular functions and the level at which they take place. For instance, if we look at BRCA1, we can see in one synthetic view all the cellular functions in which the BRCA1 BARD1 complex is involved. So, let's recap everything we've just said. The fact that biological sequences are the product of an evolutionary process comes along with many benefits for us. It allows us to consider every sequence as the product of natural selection, thus making it possible to associate sequence conservation and functional relevance. The tools of choice to do this analysis are multiple sequence alignments and phylogenetic reconstruction. Conservations revealed by multiple sequence alignments make it possible to identify somatic mutations and potential drivers of a disease. When the mutated genes are not actionable, that is, when they cannot be targeted with a drug, system biology tools such as KEGG can allow the identification of alternative targets based on a pathway modeling of the cell. It is now time to say goodbye. I hope you have enjoyed these 4 weeks of introduction to bioinformatics. I'm sure you will understand that we have only scratched the surface of all the possibilities and novel therapeutic opportunities bioinformatics will bring us. Above all, I hope you have now come to realize that what we call bioinformatics is merely regular biology in which formidable machines such as sequencers and mass spectrometers or even microscopes are now interfaced with human knowledge via computers. Aside from this, it changes nothing in the way biological knowledge can be harnessed for the benefit of human health. And I wish you a lot of interest and a lot of pleasure in the rest of this series of lectures.